Okay, so we'll be doing on Bacillus. What I want you guys to remember from the previous lectures, we said there are only two bacteria that form spores: aerobic bacilli and anaerobic Clostridia. Okay, so sporogenous gram-positive bacilli. Then we have the non-bulging spores, which are the Bacillus, and then the bulging spores, which are the Clostridia. From here, let's talk about the general features of Bacillus. They are ubiquitous. Ubiquitous means that they can grow anywhere, or they can be found anywhere: the soil, dust, air, water. They are generally motile with peritrichous flagella. Okay, so peritrichous is where we have a lot of flagellae emanating from the bacteria itself. And then we have two types of bacillus that are important to us: Bacillus anthracis and Bacillus cereus. So Bacillus anthracis is non-motile, and it is the one that causes anthrax. I'm sure you've heard of anthrax. It's a quite common disease, mostly in animals, but it can also occur in human beings. Then Bacillus cereus is a bacteria that causes food poisoning. We're going to see. They are aerobes and facultative anaerobes. So they can depend. They can be aerobes or facultative anaerobes. So this means that they can do with oxygen or without oxygen. And they are catalase positive. Okay. Remember catalase, we said it breaks down hydrogen peroxide to water and oxygen. Those are general features. So we start with Bacillus anthracis. It is zoonotic, meaning it affects both human beings and animals. The word anthrax means coal. Okay. It forms something we known as black Escher. Okay, so this is very high yield. If you see in a question black Escher, just know that is Bacillus anthracis. Then this is the history of Bacillus anthracis. In 1849, it was first seen in the microscope, became a communicable disease, isolated in 1876. Then in 1881, they formed the first attenuated vaccine. Morphology: gram-positive bacteria, non-acid fast bacilli. So acid-fast bacilli, I want you guys to remember there are two types. So acid-fast bacilli, the two types of bacteria you'll remember is Mycobacterium tuberculosis and Nocardia. Okay, we're going to do Nocardia and the branching filament gram-positive bacteria. Then it is non-motile, non-bulging. It has a central oval spores. It forms central oval spores. Who remembers the function of spores? What did we say the function of spores is?
It helps them to survive in adverse conditions.
Yes, good. So such as heat, chemicals, okay? And then the capsule. So it is capsulated. And what I want you to note here is the only bacteria that has a polypeptide capsule. Okay? Remember the other ones had a... What was the capsule for the other bacteria? What was the main ingredient? It is the only bacteria with a polypeptide capsule. I remember I also told you this when we were talking about structure of bacteria. What is the normal structure of what is a capsule made of? Who remembers? You see, no. What is a polysaccharide? Told you the answer. So the capsule is made of polysaccharide. Okay? It is made of polysaccharide. But in Bacillus anthracis, it is made of polypeptide. So take note of that. So if you see a question, polypeptide capsule, that is Bacillus anthracis. surrounds bacillary chains in tissues, seen only under 10 to 25% CO2, serum, starch, albumin, and charcoal. The main thing I want you to know is the polypeptide capsule. And then the spores, these are never seen in tissues, only in culture. So we have water and 2% sodium chloride, or in environment, calcium chloride inhibitory. So this is how we're going to see the spores. Okay, so the main thing here you have to know is it's made up of a polypeptide capsule and the spores. And then we have McFadden reaction. This is not quite common. So this is not high yield. GMSA are also not high yield. GMSA is mainly used to stain parasites, but it can also be used here. It will appear purple and the capsule will appear red, but this is not done in the lab. You won't, even when you do your practicals, you're not going to use GMSA stain. And then something we call bamboo stick appearance. Okay, this is buzzy, very buzzy for Bacillus anthrax. It has a bamboo stick appearance. So end-to-end arranged long chains of bacilli in the culture. I think I have a picture. Can you see here? They have bamboo. You know how bamboo looks like? If I was to draw, they look like this. Okay. That is what we call the bamboo stick appearance. Okay. And then on the culture, we have something called as medusa head appearance. So this is also something you should know. So as you can see here, this is Medusa. If you know the history of Medusa, she had snakes on her head. So this snaky appearance is what we call bamboo. Sorry, is what we call Medusa head appearance. Basically, these are colonies. If you were to see these, they show a halo of projections. Okay. Now that halo of projections is what we call Medusa head appearance. Okay. So at low power lengths, the edge long interlacing chains of bacilli. As you can see, these chains, they're interlacing, so they're intertwining. That's what we call Medusa head appearance. Then gelatin stab culture, this is also not done, but you're going to see inverted feed tree, as you can see here in this picture. Selective media that we use, PLET, poly-B lysoenzyme, EDTA, and thallus acetate. Then strings of pearls grown on penicillin agar, the bacilli enlarged, and their spherical chains, as you can see here. Okay. Gamma phage, they're susceptible, but for Bacillus cereus, it is resistant. GMS, acid only. So this is gram stain, catalase and nitrate positive. So what I want you to get from this slide is the Medusa head appearance, and it is both catalase and nitrate positive. Okay. As I had shown you, the Medusa head appearance. And then resistance. So vegetative, it is killed at 60 degrees for 30 minutes. For the spores, so the vegetative are the bacteria, not the spores. For the spores, they resist dry heat at 140 degrees for 1 to 3 hours. Boiling kills them at 10 minutes, viable in the soil for very many years. Okay. So that's how the animals get them. So when they consume, because they stay viable in the soil for very many years. They are destroyed in 4% potassium permanganate and autoclaving. And then there's a process called dackering where we use 2% formaldehyde at 30 to 40 degrees for 20 minutes. Okay, so the most important thing here is for you to know that this presence of spores makes it resist dry heat at 140 degrees. Now we go to the virulence factors of Bacillus anthracis. Number 1, we have the capsular antigen, which is made up of polypeptide, hapten, and this is virulence. What did we say is the function of a capsule? Someone tell me. What did you say is the function of a capsule in all the organisms that we have gone through? and that we are going to go through. I've seen someone has said adhesion, yes, but the main function of a capsule is it protects the bacteria against phagocytosis. So phagocytosis is the process where a macrophage is going to swallow up the bacteria. That is usually the main function, but adhesion is also a function of the capsule. But for adhesion, it's mainly the slime layer. Do you remember? We said the slime layer, where it adheres to surfaces. So that's why you're going to have biofilm formation in things like indwelling catheters, okay? And then we have the cell wall antigen, which is made of polysaccharide, somatic antigen, which is heat labile protein, protective antibody, okay? So now we go to the toxins. This is the most important. I want you to know the names. We have OF, which means edema factor, PA, protective antigen, and LF, lethal factor. All these make up the anthrax toxic complex. Okay? They have no effect individually, so they have to be a complex for it to work. Okay? So edema factor, it activates adenyl cyclase, leading to accumulation of cyclic AMP, causing edema. So if I were to show you a picture, if you see this picture, this is what, remember I called it the black Escher. So if you were to look at here, there's some necrosis taking place here, basically the dark part. And then if you're to see around here, let me draw for you guys. If you're to see, here is the black part, which is the black Escher. And then this part that I've highlighted, the part surrounding that necrotic tissue. So the reason for that is because of the edema factor. Can you see? The surrounding area, that black part, is because of the edema factor. Okay? And then the protective antigen bind target cells and provide binding sites for the edema factor and the lethal factor. Then the lethal factor, basically it kills cells. So the mechanism of which it kills cells is it usually inhibits cell signaling. So the cell cannot communicate. And once it happens, apoptosis will happen. Basically, the cell will rupture. Okay, and these cells are the immune cells. So for example, the immune cells have come to the site of action and this toxin has been released. Remember, these are exotoxins. They have been released. What will happen is there won't be proper cell signaling. So the cell cannot signal and say, okay, we need to destroy this. So apoptosis will happen and the cell will rupture. Okay. So this accumulation of CMP is what will cause the edema. Basically, the edema means that increase in fluid. That's why, as you can see here, it is slightly bumpy because there's an increase in fluid. Okay. And funny thing is, these things are actually painless. Okay. The necrotic area, the black area that you see there, it's usually painless. Okay. Then this is usually the pathogenesis. From animals, they shed, goes to the soil. As it said, it can stay active in the soil because of the spores. Then the humans ingest them. Then it goes like a cycle. There are two types of diseases that Bacillus anthracis cause that you have to know. We have the cutaneous manifestation and the pulmonary manifestation. So cutaneous, this deals with skin. We call it cutaneous anthrax. This, as you can see, it can occur on the face, the neck, the hands, and the back. We call it hard Potter's disease. The reason it's called that is because it was mainly seen in people who had, sheep, goats, cattle, okay? The farmers, abattoirs, and those who handle carcasses. Then the skin abrasions caused by the spore lead to a papule. So a papule is a raise in a bump on the skin. and then it becomes a vesicle. Once fluid starts accumulating, it becomes a pustule, causing inflammation and necrosis. Necrosis is now it starts becoming black. Finally, it becomes an eschar, as you can see here. Maybe I can erase it. Okay. As you can see here, there is necrosis and the surrounding area is edematous. Okay, that's for the cutaneous manifestation. And remember, it is painless. Pulmonary manifestation, this means that it involves the lungs. What happens is when you inhale the spores, okay, from handling of wool or the carcasses of these animals, that's why it's called wool sorter's disease. So the people who'd sort wool after, collecting the wool, they'd get these manifestations. So what happens is after ingesting the spores, you develop flu-like symptoms, okay? So you start coughing, sneezing, then you start getting fever, and then you get bronchopneumonia. It goes and develops pneumonia. So hemorrhagic bronchopneumonia. So meaning there'll be blood involved, okay? And then septicemia. So this septicemia happens when the bacteria enters the blood, and then you get septicemia, okay? And then this increases the rate of mortality. So you can compare between cutaneous and pulmonary. Which one is more likely to kill you? Cutaneous, when you get cutaneous anthrax or pulmonary anthrax?
To be pulmonary.
Pulmonary, very good. And then we have the intestinal. This is quite rare. I've never even seen it. But due to improperly cooked meat, you can get bloody diarrhea, and this can increase the mortality. But this is actually very rare. When you're asked diseases that Bacillus anthrax cause, the first one should be the cutaneous one, then pulmonary. As you can see here, anthrax spores are inhaled. They enter the lungs and travel to the alveolar spaces. Spores are transmitted through the lymph node to the mediastinal lymph nodes, where they make toxins that are deadly. And you see here, so when they reach the lymph nodes, they start releasing their toxins. And which toxins are these? Edema factor, protective antigen, and lethal factor. Okay. Hope you've understood. So this is usually the lab diagnosis. For our samples, we have the swab because you have pulmonary involvement, pus, fluid, sputum, blood to see if there's the presence of the bacteria. And then gram stain, what are you going to see? So the chains of large gram-positive bacteria, okay? And then when you culture, medusa head colonies, animal inoculation. So when you culture in a mouse, it causes death in 48 hours at blood gram-positive bacteria. Serology, this is not usually done. It's usually done by biochemists. And then PCR is to detect the antigens of these. Okay, but the most important here is that it is gram-positive bacteria, and when culturing, you're going to see medusa head colonies. Prophylaxis, improve factory hygiene, sterilize animal products, suspect animal carcasses, lime burial. So they're usually buried. You don't eat them because you can get infected. Then active immunization of animals. Okay, not safe for human. Then human vaccination. Okay, especially those who are in contact with these animals. Okay, we are done with Bacillus anthracis. Do you have any question? Okay, we go to Bacillus cereus. So for Bacillus cereus, I want you to get three things, only three things. It causes food poisoning. How? Normally what happens is, remember I said Bacillus produces spores, okay? Now what will happen is the ideal situation for Bacillus cereus that you're going to see maybe in the exam is, I think I have a diagram here. Yes, rice. So you're going to cook rice. And remember, we said the spores are going to survive heat. Okay. So after cooking, here is your rice. So you're going to, let's say you've eaten your rice a certain portion and then you've left the rice overnight. Maybe you've stored it at warm temperatures. Let's say room temperature. What is going to happen is, remember there are spores in this rice. So when you reheat it again, when you want to cook it again, so you've stored it at room temperature, then the next morning you want to reheat it and eat it again. There are two ways it's going to cause disease. There is the ingestion of the toxin, okay? And there's the ingestion of the spores, okay? Remember, these spores will produce the enterotoxin. Then you're going to ingest those enterotoxin that will now cause disease, okay? The two types of diseases it causes is the emetic type and the diarrheal type. For emetic, emesis means vomiting. Basically, you're vomiting, okay? Nausea and vomiting occurs after eating Chinese fried rice, okay? So it's similar to staphylococcus. Remember how we said staphylococcus causes food poisoning? Quite similar, 1 to 5 hours. So if you see something that you've eaten, then you take 1 to 5 hours, you start developing symptoms. You know you have ingested the toxin. What is responsible here is the toxin, okay? And then we have the diarrheal type, which takes quite longer because by the time food reaches the small intestine, you have abdominal pain, diarrhea. Vomiting is not common, okay? And the diarrhea here is non-bloody, okay? It's watery diarrhea. And it, as you can see, from 8 to 16 hours, okay? That's what I want you to remember. It is caused by reheating rice. You ingest the spore. The toxin causes you to vomit. Then after 8 to 16 hours, you have the diarrheal type. So it's different types of toxins. The toxin here, it's known as cerulide. Let me write it for you guys. Sorry, I didn't include it in the slides. Cerulide toxin. Cerulide toxin. Okay, it's the one that causes the emetic type that will cause you to vomit 1 to 5 hours after eating. Okay. Management of this is usually supportive because, you know, once a toxin has been released, you can't give like antibiotics compared to Bacillus anthracis. You could give antibiotics to kill the bacteria itself. But once the toxins have been released, it's quite hard to manage that. So this is just a diagram. So rapid deep fry kills bacteria and not the toxins. Then you ingest the toxins, acute vomiting 2 to 4 hours after ingestion. Inadequate reheating, the bacteria will remain. Ingestion of spores or viable bacteria. It multiplies in the gut, causing enterocolitis or the watery diarrhea. Okay. That's all I want you to remember for Bacillus cereus. These ones are not usually asked, but for the sample, food, feces, vomiters. Okay. Yes. I'm done with Bacillus. Do you have any question?